Welcome, dear students. Today we are going to discuss about bioprocesses and fermentation. Bioprocesses or fermentation technology is an important component of most old and new biotechnological processes and normally involve complete living cells, either microbial, mammalian or plant cells, organelles or enzymes as the biocatalysts. It aims to bring out specific chemical or physical changes in biochemical materials derived from the medium. In order to be viable in any specific industrial context, bioprocessing must possess advantages over competing methods of production, such as chemical technology. In practice, many bioprocessing techniques will be used industrially because they are the only practical way in which a specific product can be made. Example, vaccines, antibodies. Biochemical engineering covers the design of vessels and apparatus suitable for performing such biochemical reactions or transformations. The very beginning of fermentation technology, or as it is now better recognized as bioprocess technology, were derived in part from the use of microorganisms for the production of foods such as cheese, yogurts, sauerkraut, fermented pickles and sausages, soy sauce and other oriental products, and beverages such as beers, wines and derived spirits. In many cases, the present-day production processes for such products are still remarkably similar. These forms of bioprocessing were long viewed as arts or crafts, but are now increasingly subjected to the full array of modern science and technology. Paralleling these useful product formation was the identification of the role microorganisms could play in removing obnoxious and unhealthy wastes, which has resulted in the worldwide service industries involved in water purification, effluent treatment, and solid waste management. Bioprocessing in its many forms involves a multitude of complex enzyme-catalyzed reactions within specific cellular systems, and these reactions are critically dependent on the physical and the chemical conditions that exist in their immediate environment. Successful bioprocessing will only occur when all the essential factors are brought together. Although the traditional forms of bioprocess technology related to foods and beverages still represent the major commercial byproducts, new products are increasingly being derived from microbial, mammalian, and plant cell fermentations, namely the ability to overproduce essential primary metabolites such as acetic acid, lactic acid, glycerol, acetone, butyl alcohol, organic acids, amino acids, vitamins, and polysaccharides, to produce secondary metabolites such as penicillin, streptomycin, cephalosporin, gibberellins, etc., to produce many forms of industrially useful enzymes, example, extracellular enzymes such as amylases, pectinases, and proteases, and intracellular enzymes such as invertase, asparaginase, restriction endonuclease, etc., to produce monoclonal antibodies, vaccines, and novel recombinant products like therapeutic proteins. More recently, bioprocess technology is increasingly using cells derived from the higher plants and animals to produce many important products. Plant cell culture is largely aimed at secondary product formations, such as flavors, perfumes, and drugs, while mammalian cell culture has been concerned with vaccine, antibody formation, and the recombinant production of protein molecules such as interferons, interleukins, and erythropoietin. The future market growth of these bioproducts is largely assured because, with limited exceptions, most cannot be produced economically by other chemical processes. By genetically modifying organisms to have better or unique productivities and utilizing new processing technology advancements will make it feasible to achieve additional economies in the production. The product formation stages in bioprocess technology are essentially very similar, no matter what organism is selected, what medium is used, or what product is formed. The organisms must be cultivated to form the desired products by means of physical, technical containment systems, the bioreactor, and the correct medium composition and environmental growth regulating parameters such as temperature and aeration. Optimization of the bioprocess spans both the biological and the technical systems. The proper exploitation of an organism's potential to form distinct products of defined quality and in large amounts need a detailed knowledge of the biochemical mechanisms of product formation. Bioprocessing in its many forms is catalyzed within each respective cellular system by a large number of intracellular biochemical reactions. Substrates derived from the medium are converted into primary and secondary products, intra and extracellular macromolecules, and into biomass components such as DNA, RNA, proteins, and carbohydrates. These reactions are dependent on the physical and chemical parameters that exist in their immediate environments. The same apparatus with modifications can be used to produce an enzyme, an antibiotic, an amino acid, or a single cell protein. In its simplest form, the bioprocess can be seen as just mixing of microorganisms with a nutrient broth and allowing the components to react. Example, yeast cell with a sugar solution to give alcohol. More advanced and sophisticated processes operating at large scale need to control the entire system so that the bioprocess can proceed efficiently and readily and can be exactly repeated with the same amount of raw material and inoculum to produce precisely the same amount of product. All biotechnological processes are essentially performed within containment systems or the bioreactors. Large number of cells are invariably involved in these processes, and the bioreactor ensures their close involvement with the correct medium and conditions for growth and product formation. It also should restrict the release of cells into the environment. A main function of a bioreactor is to minimize the cost of producing a product or service. We'll talk about the bioreactors. Bioreactors are the containment vehicles of any biotechnology-based production process, be it for brewing, organic or amino acids, antibiotics, enzymes, vaccines, or for bioremediation. For each biotechnology process, the most suitable containment system must be designed to give the correct environment for optimizing the growth and metabolic activity of the biocatalyst. Bioreactors range from simple stirred or non-stirred open containers to complex aseptic integrated systems involving varying level of advanced computer controls. Bioreactors occur in two distinct types. In the first instance, they are primarily non-aseptic systems, where it is not absolutely essential to operate with entirely pure cultures. Example, brewing, effluent disposal system. While in the second type, aseptic conditions are a prerequisite for successful product formation. Example, antibiotics, vitamins, polysaccharides, and recombinant proteins. This type of process involves considerable challenges on the part of engineering construction and operation. The physical form of many of the most widely used bioreactors has not altered much over the past 40 years. However, in recent years, novel forms of bioreactors have been developed to suit the needs of specific bioprocesses, and such innovations are finding increasingly specialized roles in bioprocess technology. In all forms of fermentation, the ultimate aim is to ensure that all parts of the system are subjected to the same conditions. Within the bioreactor, the microorganisms are suspended in the aqueous nutrient medium containing the necessary substrate for the growth of the organism and required product formation. All nutrients, including oxygen, must be provided to diffuse into each cell, and waste products such as heat, carbon dioxide, etc. must be removed. The concentration of the nutrients in the vicinity of the organism must be held with a definite range, since low values will limit the rate of organism metabolism, while excessive concentrations can be toxic. Biological reactions run most efficiently within optimum ranges of environmental parameters. And in biotechnological processes, these conditions must be provided on a micro scale so that each cell is equally provided for. The difficulty of achieving these conditions in a population as a whole becomes evident when one considers the huge scale of many bioreactor systems. It is here that the skills of the process or biochemical engineer and the microbiologist must come together. Fermentation reactions are multi-phase, involving a gas phase containing nitrogen, oxygen, and carbon dioxide, one or more liquid phase containing aqueous medium and liquid substrate, and the solid microphase, which include the microorganisms and possibly solid substrate. All phases must be kept in close contact to achieve rapid mass and heat transfer. In a perfectly mixed bioreactor, all reactants entering the system must be immediately mixed and uniformly distributed to ensure homogeneity inside the reactor. To achieve optimization of the bioreactor system, the following operating guidelines must be closely adhered to. The bioreactor should be designed to exclude entry of contaminating organisms as well as to contain the desired organisms. The culture volume should remain constant, that is, no leakage or evaporation should occur. The dissolved oxygen level must be maintained above critical levels of aeration and culture agitation for aerobic organisms. Environmental parameters such as temperature, pH, etc. must be controlled and the culture volume must be well mixed. The standard of material used in the construction of sophisticated fermenters is important. Fermentation technologists seek to achieve a maximization of culture potential by accurate control of the bioreactor environment. But still, there is a great lack of true understanding of just what environmental conditions will produce an optimum yield of organism or product. Organisms with large cell size, such as animal cells compared to bacteria, have complex demand for nutrients and a lower growth rate. On the other hand, their ability to produce complicated proteins is more. Successful bioprocessing will only occur when all the specific growth-related parameters are brought together in order to improve and optimize the process. For successful commercial operation of these bioprocesses, quantitative description of cellular processes is an essential prerequisite. The two most relevant aspects, that is, the yield and productivity, are quantitative measures that will indicate how the cells convert the substrate into the product. The yield represents the amount of product obtained from the substrate, while the productivity specifies the rate of product formation. To understand and control a fermentation process, it is necessary to know the state of the process over a small time increment, and further to know how organism responds to a set of measurable environmental conditions. Process optimization requires accurate and rapid feedback control. The computer has become an integral part of most bioreactor systems. However, there is a lack of good sensor probes that can allow online analysis to be made on the chemical components of the fermentation processes. A large worldwide market exists for the development of new rapid methods monitoring many reactions within a bioreactor. In particular, the greatest need is for innovatory microelectronic designs. While endeavoring to improve existing process operations or design, it is often advisable to set up mathematical models of the overall system. A model is a set of relationships between the variables in the system being studied. Such relationships are usually expressed in the form of mathematical equations, but can also be specified as cause-effect relationships, which can be used in the operation of the specific processes. The actual variables involved can be extensive, but will include any parameter that is of importance for the process and can include pH, temperature, substrate concentration, agitation, feed rate, etc. Bioreactor configurations have changed considerably over the last few decades. The original fermentation system was a shallow tank agitated or stirred by manpower. From this has developed the basic aeration tower system, which now dominates industrial usage. As fermentation systems were further developed, two design solutions to the problems of aeration and agitation have been implemented. The first approach uses mechanical aeration and agitation devices with relatively high power requirements. The standard example is the centrally stirred tank reactor, that is CSTR, which is widely used throughout the conventional laboratory and industrial fermentations. Such bioreactors ensure good gas-liquid mass transfer, have reasonable heat transfer, and ensure good mixing of the bioreactor contents. The vertical shaft of the CSTR will carry one or more impellers depending on the size of the bioreactor. A broad range of impellers have been investigated for stirring and creating homogeneous conditions within the bioreactor. The impellers are usually spaced at intervals equivalent to one tank diameter along the shaft to avoid a swirling type of liquid movement. The 6 flat-bladed Rushton turbine impellers are used in the majority of bioreactors, and normally 3 to 5 are mounted to achieve good mixing and dispersion throughout the system. The function of the impellers is to create agitation or mixing within the bioreactor and to facilitate aeration. The primary function of agitation is to suspend the cells and nutrients evenly throughout the medium, to ensure that the nutrients including oxygen are available to the cells and to allow heat transfer. Most industrial organisms are aerobic, and in most fermentations the organisms will exhibit a high oxygen demand. Since oxygen is sparingly soluble in aqueous solutions, aerobic fermentations can only be supported by vigorous and constant aeration of the medium. The second main approach to aerobic bioreactor design uses air distribution with low power consumption to create forced and controlled liquid flow in a recycle or loop bioreactor. In this way, the contents are subjected to a controlled recycle flow, either within the bioreactor or involving an external recycle loop. The stirring has been replaced by pumping, which may be mechanical or pneumatic, as in the case of airlift bioreactors. The centrally stirred tank reactor consists of a cylindrical vessel with a motor-driven central shaft that supports one or several agitators, with the shaft entering either through the top or the bottom of the vessels. The aspect ratio, that is, height to diameter ratio of the vessel is 3 to 5 for microbial system, while for the mammalian cell culture the aspect ratios do not normally exceed 2. Sterile air is sparged into the bioreactor, liquid below the bottom impeller by way of a perforated ring sparger. The speed of the impellers will be related to the degree of fragility of the cells. Mammalian cells are extremely fragile when compared to the most microorganisms. In many of high-value processes, the bioreactors will be operated in a batch manner under aseptic monoculture. The initial culture expansion of microorganisms will commence in the smallest bioreactor, and when growth is optimized, it will then be transferred to a large bioreactor, and so forth until the final operation bioreactor. Throughout such operation, it is imperative to maintain aseptic condition to ensure the success of the process. Bioreactors are normally sterilized prior to inoculation, and contamination must be avoided during all subsequent operations. If contamination occurs during the cultivation, this will invariably lead to the process failure, since more often the contaminant can outgrow the participating monoculture. The number of distinct types of bioreactors is quite limited when measured against the wide range of productive processes and the varied biological systems involved. In industrial practice, the CSDR now occupies a dominant position and is virtually the only bioreactor designed used in full-scale bioprocessing. Large amounts of organic wastewaters from domestic and industrial sources are routinely treated in aerobic and anaerobic systems. Activated sludge processes are widely used for the oxidative treatment of sewage and other liquid. Such processes use batch or continuous agitated bioreactor systems to increase the entrainment of air to optimize oxidative breakdown of the organic materials. These bioreactors are large and for optimum functioning will have several or many agitator units to facilitate mixing and oxygen uptake. They are widely used in most municipal sewage treatment plants. Anaerobic bioreactor or digesters have long been used to treat sewage matter. In the absence of free oxygen, certain microbial consortia are able to convert biodegradable organic materials to methane, carbon dioxide, and new microbial biomass. Most common anaerobic digesters work on a continuous or semi-continuous manner. An outstanding example of the methane generation is the Chinese biogas program, where millions of family-sized anaerobic bioreactors are in operation. Such bioreactors are used for the treatment of manure, human excreta, etc., producing biogas for cooking and lighting and the sensitization of the waste, which then becomes an excellent fertilizer. In almost all fermentation processes performed in a bioreactor, there is generally a need to measure specific growth-related and environmental parameters, record them, and then use the information to improve and optimize the process. Bioreactor control measurements are made either through an online or an offline manner. With an online measurement, the sensor is placed directly within the process stream, whereas for offline measurement, a sample is removed aseptically from the process stream and analyzed. Bioreactor processing is still severely limited by a shortage of reliable instruments capable of online measurements of important variables such as DNA, RNA, enzymes, and biomass. Offline analysis is still essential for these compounds, and since the results of these analyses are usually not available until several hours after sampling, they cannot be used for immediate control purposes. However, online measurement is readily available for temperature, pH, dissolved oxygen, and carbon dioxide analysis. The continued discovery of new products such as therapeutic drugs from microorganisms and mammalian cells will continue to depend on the development of innovative exploratory culture systems which encourage the biosynthesis of novel compounds. New miniaturized computer-controlled incubator systems with automated analysis unit are now available as a single units that can perform hundreds of experiments simultaneously, thus producing a wealth of data in a short time to facilitate optimum fermentation conditions for product formation. A new, quite novel approach involving combinatorial biology generates new product from genetically engineered microorganisms. DNA fragments or genes derived from unusual microorganisms that are not easily cultivated, that is, recalcitrant microorganisms, can be transferred into easily cultivated or surrogate microorganisms, and the resulting mixing and matching of genes encoding biosynthetic machinery offer the opportunity to discover new or modified molecules or drugs. This could be of great significance in antibiotic discovery. While most high-value biotechnological compounds such as antibiotics and therapeutic proteins are produced in monoculture under strict conditions of asepsis, there are now new avenues of research exploring product formation from mixed culture systems. Such systems may well produce different patterns of metabolites or indeed novel metabolites as a result of interaction that can occur between competing microorganisms. Because of the complexity of these mixed organisms processes, they have all but been ignored by the scientific community. Monoculture under aseptic condition is totally unnatural and rarely, if ever, occurs in nature. The norm is for microorganisms to exist together in the environment and to compete and respond to substrate availability and to the prevailing environmental conditions. If we talk about scale-up, most biotechnological processes have been identified at laboratory scale, and ultimately commercial success will be dependent on the ability to scale up the process first from laboratory to pilot plant level and then to full commercial scale. The achievement of successful process scale-up must fit within the range of physical and economic restraints. The identification of some of the controlling parameters can usually be made with laboratory scale bioreactor, 5 to 10 liters in capacity, and then moved to pilot scale level. A pilot plant is in reality a large-scale laboratory which has been designed to give flexibility for equipment accommodation and adaptability for process operation. Pilot plant bioreactors range from 100 to 10,000 liters total volume, and the larger pilot bioreactor can on occasion be used as production units. The management of scale-up requires high capital investment in mixing and aeration, in monitoring and control devices, and in stringent maintenance of sterility. In conclusion, bioprocesses and fermentation technologies play an important role in various industries, ranging from pharmaceuticals and food production to biofuel and environmental management. Bioprocessing offers sustainable and eco-friendly alternatives to traditional chemical processes, reducing environmental impact and resource consumption. Moreover, advancements in bioprocess engineering, automation, and optimization have led to increased yields, reduced production cost, and improved scalability. The ability to manipulate microbial strains through genetic engineering has opened up new possibilities for tailoring bioproduction processes to meet specific requirements. In summary, bioprocess and fermentation technologies represent a dynamic and evolving field with far-reaching implications for diverse industries, paving the way for a more sustainable and efficient future in the production of essential compounds and materials. With this, we will conclude today's lecture. Thank you.